of research experiences. From the University of Montana, rising senior, Madeline Bradbury. And she worked in the McDonough lab.
Hi, everyone. Thank you for coming out today. So my name is Madeline Bradbury. I had the pleasure of working in the McDonough lab this summer on a pilot study assessing microglial activation in the hippocampus. So for a little bit of background information, studies on preterm infants are limited to human observational studies. And what we know from these is that they are more likely to show cognitive, motor, behavior, and language deficits. They're also at a higher risk of systemic inflammation, which can negatively impact neurodevelopment. These studies are also looking at diet and the effects of human milk in both preterm and term infants, and these have found that it increased growth and performance outcomes in both groups. Now, observational studies are great. They can tell us a lot, but they can't tell us anything about the mechanism of how diet is impacting development. So this is where our study comes in. And preterm infants are a very vulnerable population. They're heterogeneous. A lot of factors can affect their outcomes. This could be degree of prematurity, location, socioeconomic status. Animal models help mediate some of these variables and let us look into the mechanism more. So for this study, piglets were chosen specifically because they have similar anatomical features to humans. Their white to gray matter ratio is similar to humans, and they have similar anatomical features. So for this study, we had two treatment groups, one that was fed human milk and one that was fed infant formula, starting at 2 days, going until they were 30 days old. In both groups, the first 2 days, they were fed colostrum from the sow to help establish their immune system. Now, it's important to note that this project is a joint project with the University of Idaho, and they are looking at the integrity of the intestinal barrier. So what we would expect to see is that in the piglets that were fed a human milk diet, we should see a less permeable intestinal barrier. And then looking at the immune system, less systemic inflammation and less neuroinflammation. And this would allow for optimal neurodevelopment in this group. So moving on to what I did this summer, which I absolutely loved, I looked at the microglia and the hippocampus. We chose microglia because these are the innate immune cells of the central nervous system, and they can help indicate neuroinflammation because they will respond to any sort of damage, infection, or inflammation in the brain. And when they do this, you'll see an acute change in morphology. So you can see in this picture right here, this is a surveying microglia. They've got these long processes, and there's very few of them. Whereas when they're activated, they take on this more amoeboid-like shape. They have shorter processes and much more of them. We'll also see an increase in proliferation and an increase in microglial density in the tissue that's affected. Now, we chose the hippocampus specifically because it's involved in learning, memory, and spatial navigation, and these are impacted in preterm infants. So my hypothesis for this study is that in the piglets that were fed a human milk diet, we should see less inflammation and therefore less activated microglia, less proliferating microglia, and an overall decreased microglial density in the tissue. And this is me and Gino working at the cryostat, which was a lot of fun. So as you can see here, the hippocampus was dissected out of the piglet brains. It was frozen in this medium. So this is the hippocampus right here. The blade is right here, and they were cut into very small sections and then picked up on a slide. Once they're on the slide, they underwent a round of IHC or immunohistochemistry. This is indirect antibody staining. So with this, you have a primary antibody that will recognize a specific cell type or marker, and then a secondary antibody that has a fluorescent tag that will bind to the primary antibody. And this helps us visualize and actually see the cells under the microscope. And for this project, we use IBA1, which is this nice green color, to stain microglia, Chi67, which will mark proliferating cells, and DAPI, which will stain DNA so we can see all of the nuclei. So in this picture right here, you can see these are proliferating microglia, likely post-mitotic sister cells. Then once I went through the IHC, I've got it on the slide, I went to the Marianas to do some imaging. So in this section, I chose an area around the dentate gyrus. Once I had that area selected, using the stereology module on the computer, it randomly selected sampling sites that I then went in and imaged further at a higher magnification. And it's important to remember here that even though they are very thin slices of tissue, they still have depth. So when you're going and you have to image at several planes to make sure you're capturing all of the cells in that area. So again, what we would expect to see in these results is that in the piglets that were fed a human milk diet, we should see less microglial density and less microglial proliferation. So looking at this first graph, this is comparing human milk and infant formula treatment groups. For IBL1 positive cells are the microglia and the rostral dentate gyrus. And here we can see that there was no statistical significant difference between these two groups. Then looking at just proliferating cells in the two groups, again, we see no significant difference between the two treatment groups. And finally, looking at microglia that were proliferating, there was no statistical significance between these two groups. Now, what this means, there was no difference that I found between the groups on my summer project but it's important to remember that this is a preliminary study. Right now, we're working with an N of 3, meaning there's 3 piglets in each treatment group. And based on a power analysis, to be confident in these results, we would need at least an N of 14. As I was doing this project, I also only imaged 1 slide per piglet, and we would need to increase this number to be more confident in these results. But they're also not terribly surprising. We know that infant formula is very well tolerated, and these findings were consistent with previous student work looking at the prefrontal cortex, and then work that I did looking at the unical dentate gyrus. Moving forward, we want to quantify different regions in the brain and look at morphology studies. So I mentioned before, you've got kind of this range of activation with the microglia, and just looking at them, it can be hard to tell how activated they are. So using 3D morph, you can actually characterize how activated they are, and we can see if there's differences between those two treatment groups. And then looking further, we'd like to look at proteins, transcriptomics, and assess white matter development by looking at myelination and oligodendrocyte proliferation and maturation. Now, I want to say a huge thank you to the McDonough lab. You've been so welcoming this summer. I absolutely loved working there. I want to say thank you to Dr. Richard Ellenbogen and Dr. McDonald and everyone who made this program possible. It's been a truly incredible experience.